I think we can go ahead and get started. First of all, good afternoon, everybody. My name is Paul Grossfeld, and there will be a much more formal greeting and opening remarks this evening, starting at 6, by the way. In case you don't know, we had to move it back by about an hour for the poster session now to start at 6. I think, Lindsay, correct me if I'm wrong, dinner will start at 7, is that correct? So, and the reason for that is the biopsies got a little bit delayed, or the schedule had to be changed a little bit. So, but I will informally say now, thank you, all of you, for coming. I was telling someone earlier, it's like planning a wedding. There's been so many months of anticipation leading up to today, and it's all now arrived, so we're all very excited. We're going to start out this afternoon's early bird session with Dr. Marie Dell'Aquila, and I think Marie and I are both a little bit embarrassed to acknowledge how long we've known each other for, correct? But she doesn't look any older than 20 years ago when I first met her, so I, on the other hand, that's a different story. But Dr. Dell'Aquila is the director of the Cytogenetics laboratory at UCSD. She has, I think, probably seen more abnormal chromosome 11s under the microscope than anybody in the world. And so in keeping with the tradition, I always like to start out the conference with sort of a basics background on genetics. And I think one of the things that's always fascinating about having Dr. Dell'Aquila talk is every 2 years there have been so many advances in the technologies that there's always going to be new and exciting information to hear about. And I think by hopefully getting everybody on the same page from the beginning, you're going to be hearing a lot about chromosomes and breakpoints and megabases and all that kind of stuff. And I think Dr. Dell'Aquila will set us straight on all that. So I will have a little more formal discussion but later this evening, but we're very fortunate to have donated from the Sulpizio family here in San Diego wine from their personal vineyard up north from here. And so I have a little present to you, Marie, as a token of appreciation, and thank you so much for being part of this conference and being part of our family. I forgot to bring a pointer too. Okay. Does anybody by any chance have a laser pointer? That's one thing I forgot to bring among probably many other things. Are you going to be at the podium or the computer? Because you can use you can use the mouse, or yeah.
It's a long time. Anyway, I have a few slides to show to you, and I'm happy to answer any questions. If you have something and you want to interrupt me, you're going to grab my attention, and I'm happy to answer questions as we go along. I have mostly prepared things about the basics of cytogenetics, just to get you back to remembering of how chromosomes work, what we do to analyze them in the laboratory, what kind of structural changes they can undergo that we diagnose in our patient population. This is just a slide of a metaphase spread. These are chromosomes from one cell. And again, chromosome analysis requires that cells are actively growing. So we get cells to either naturally growing or we do a little kickstart to get them to grow. And then we have some manipulation of the cell cultures so that we can get the chromosomes to condense so they can be visualized and banded and then analyzed. Okay, so why do we study chromosomes?
Is it there? Let me see if I can.
Here it is.
Oh, there you found it. Okay, it's fine. It will. Yeah, it's there. It is.
Okay,
it's just.
It's just. Okay, so chromosome disorders are one of the major categories of genetic disease, responsible for greater than 100 identifiable syndromes, and that number has actually grown a bit in the last 5 to 10 years as we started doing more molecular techniques, looking at losses and gains in chromosomes that are too small for us to see with a microscope. actually more common than all the Mendelian disorders added together. So Mendelian disorders would be things that you can inherit from one parent and the other carrying mutations in the same gene. Approximately 1% of live births have chromosomal abnormalities and about 2% of pregnancies. This is what's referred to as a karyogram, or an array of G-banded chromosomes. So they're stayed with a particular dye called visa, which is why it's called G-banded. And there's a banding pattern that's typical for each chromosome. The chromosomes are arrayed basically by size, starting with chromosome 1 being the largest. And on the bottom, the very pale small ones go through 22. The last two are the sex chromosomes. There's an X and a Y, so this is a male. When we do chromosome analysis, the spreads that we look at, the chromosomes that we see, can be of varying lengths, depending on how much the chromosomes are against. So when we look at a non-dividing cell under a microscope, and have a nucleus that contains the DNA. The DNA is organized in a structure called chromatin. So it has nucleic acids and some proteins that organize its structure. And it's basically unwound. If you were to take a bolt of yarn and just, separate it all. As chromosomes develop through cell division, they condense to form this discrete unit. This is the basic way of warehousing the genes that are in the DNA. So they're packaged into chromosomes that the cell can count 12 genes. And we have 46 chromosomes. There are pairs 1 through 22, and then the two sex chromosomes. The 1 through 22 are referred to as autosomes, and then the sex chromosomes, X and Y are XX. This particular spread. A mic for me? Okay, thank you. Let's see if I put this on and try not to walk too much. Is that working?
Put it closer.
Let me put it. Let's get on this side here. Does that help? Is that louder? Too loud? Okay. All right. So because chromosomes can be variable length and condensation, what we try to do is synchronize the cells, get them all growing at the same rate so that we can trap the dividing cells where the chromosomes are a little bit longer. This is referred to as high-resolution chromosome analysis. So these are high-resolution chromosomes, and you can get approximately 500, 600 bands that you can visually see when you do an analysis at this length at the microscope. Okay, so high-resolution karyotyping, the advantages being we can look at the whole genome when we do a chromosome analysis. It's hard to think of it, but you are actually looking at the entire genome when you look at the 46 chromosomes. It's relatively low cost, and I'll say that's relative because karyotyping is considered to be quite expensive. But certainly the molecular techniques that we now have available to us are even more expensive. The disadvantages are that it's very labor intensive. So every time chromosome analysis is done, it's one individual looking in the microscope for several hours to do one patient case. And beyond that, there's the additional review, the imaging, and before that is all the cell culture. And cell culture, depending on the kind of sample, if we're going blood or we're going cells from another part of the body, can take up to a week or 2 weeks. So it's a lot of time invested in this particular technique. Lastly, the detection level is really thought to be around 5 megabases of DNA, mega meaning million. So 5 million base pairs of DNA is about the level that we can actually see a change. So something smaller And maybe if we have really good resolution, we can get down to 3 megabases, but something smaller than that, you're not going to see it at the microscope. We just, don't have that ability. So there are molecular techniques that we have to look at smaller changes. This is an example of chromosome 11. These are all chromosome 11, starting with the very end here to your left. This is low resolution chromosome 11, and for our less experienced people who are just learning to do chromosome analysis, it's easier to recognize the shorter chromosomes. As the chromosomes become less and less condensed, or where you get higher resolution, which would be all the way over here to the right, this is a very high resolution chromosome 11. And you can see as you move from right to left that you start to see more bands, more subbands, more gray bands. So the detail that you can pick up in a higher resolution chromosome is much greater. Okay. So what happens to chromosomes when things go wrong? There are lots of different structural changes that can happen and that we certainly see when we do analyses. The first category is losing part of the chromosome, so loss of genetic material. And this can happen by either, get the hang of this, a deletion where part of the chromosome is missing. So for example, in chromosome 11, where you lose that very small segment on the bottom of chromosome 11, usually at the band that's referred to as Q23 or in that region. The deletion can be either the whole end of the chromosome is missing, or sometimes it's just a little snip from within the chromosome. So these deletions where the entire end is lost are referred to as terminal deletions, where everything from that breakpoint beyond is gone. And deletions where you just have a smaller segment missing are referred to as interstitial deletions, where the end of the chromosome is still there, but the material from the middle of the chromosome is missing. And of course, we can also lose an entire chromosome, and this is referred to as monosomy. Losing a whole chromosome is a lot of material to be missing, and so most of these are not compatible with survivability, except in the case of the sex chromosome. So losing an entire sex chromosome is compatible with long-term survival, and usually minimal effect on development and mental ability as well. It's referred to as Turner syndrome or monosomy X. Okay, so we can also have gain of genetic material. This can be either in the form of a duplication, where a segment within the chromosome is present in 2 tandem copies, or you can have gain of a whole chromosome, which is referred to as trisomy. And the most common one you'd be familiar with would be trisomy 21, which is Down syndrome. And that's gain of a whole copy of actually the smallest chromosome in our karyotype. And lastly, changes we have are relocation of material. And the most common of those are translocations. And translocations are the exchange of the end segments of 2 different chromosomes. And that's referred to as a reciprocal translocation. Because 2 chromosomes are reciprocating and they're exchanging segments between the 2 of them. Now that happens in the general population, in about 1 in 600 people carry a reciprocal balanced translocation, and many of them don't even know it. The only time that it comes into play is then when they go on to have children, and sometimes as the chromosomes divide to form the gametes, the egg and the sperm that will go on into a conception for a pregnancy, as those chromosomes divide, sometimes they don't divide in a way where both the abnormal chromosomes can segregate or migrate together into the same egg or sperm. And so occasionally carriers of translocations have conceptions or children that only have one of the abnormal chromosomes, and so they're missing part of the chromosome that was exchanged
with the other derivative, and they also have a little bit extra from what that particular chromosome received from the reciprocating chromosome. So carriers of translocations, or referred to as balanced translocation carriers, can have children or have conceptions that may end up in miscarriage that are unbalanced. So we have balanced translocation carrier parents, and sometimes they can have pregnancies that are unbalanced, or have chromosomes that are missing something and have a little bit extra of something else. Okay, the other two kinds of structural abnormalities in chromosomes are inversions. Inversions occur when an entire chromosome has an internal segment that's flipped upside down. So there are two breaks that occur in the middle portion of the chromosome, and that is inverted or flipped upside down. And that actually is quite a common occurrence. There's a particular innocuous inversion that occurs on chromosome 9 that's present in about 2% of the population. We see it very frequently, and it's not associated with any abnormality. I can't say that for all inversions, but there are many that really don't cause any problems for the individual or for their offspring. Others do. It kind of depends on the location. Lastly is insertion, and that's where one segment of a chromosome moves over and inserts into the center of a different chromosome. And those are very rare. They're very hard to pick up in analysis, and they cause all kinds of problems. Okay, so I was talking about more high-resolution molecular kinds of techniques that we have now in the laboratory. And the most common one that's still in use and very helpful for looking at very small but specific changes in the chromosomes is called FISH. And FISH stands for fluorescence in situ hybridization. Fancy word. Basically, what FISH involves is taking a known segment of DNA that has a copy sequence that identifies a particular gene or a region of a gene of interest. The particular DNA probe, as it's called, is tagged or labeled with a chemical called a fluorochrome, and it's a molecule that can emit a particular light or fluorescence when it's struck with a wavelength of light at the microscope. So we take these DNA probes that have a sequence that's identical to a region that we want to look at, tag them with something that fluoresces pretty brightly under the microscope, allow them to bind to or hybridize to the DNA of our patient, and then we wash away whatever didn't hybridize or didn't bind, and look at the microscope at our chromosomes or at our nucleus and say, okay, did my probe bind? Is that region present? So it's really a way of identifying if a region is present in the chromosome, or if it's present in multiple copies, or if it's been lost. Okay, and this just diagrams the nucleus on the lower right there with a chromosome showing the fluorescent signal from a probe that we've hybridized to it to look at the presence of a particular gene. This is an example of a FISH study that, yeah, it's hard to see these probes with any kind of light. Okay, so if you look at the image, the round circle in the upper left is a nucleus where the chromatin is not condensed, so it's dispersed DNA and not a metaphase spread, so we can't see the chromosomes discretely, but the DNA is still there, and our probes can still bind to that DNA even though it's not condensed into the form of a chromosome. The chromosomes to the right and to the lower are one metaphase spread from one cell, and we have bound to it two different probes. One probe is labeled with a green fluorochrome, and that particular probe identifies the centromere of chromosome 15. The centromere is a condensed region sort of at the end or in the middle of a chromosome that is a repetitive sequence. It doesn't code for anything, any product, but it helps keep the chromosome together, and it helps when the chromosomes are dividing into daughter cells during cell division. That particular centromere is where the sort of ropes are attached to the chromosome to pull them into the daughter cells. So centromeres have very important function in cell division, but they don't code for any protein products. The red probes, and there are actually 2 of them, tag a control on the bottom of chromosome 15, which is, you can see in the lower left, is lighting up on both chromosome 15s. And then it also tags a region that is sometimes deleted in patients that have particular syndromes referred to as Angelman syndrome and Prader-Willi syndrome. So if you look at the 2 chromosomes on the bottom, you'll see that the one on the left has 3 signals. It has a green one on top. Then right beneath the green is a red signal, and then further down is another red. The top green is a centromere. The next signal is the Prader-Willi Angelman locus, and then the bottom is a control probe. The chromosome to the right of it is an abnormal chromosome, and you can see that it's lighting up with the green probe, which says the centromere is there. It is a chromosome 15. And it's lining up with the control probe toward the bottom, but right beneath the centromere there is no red signal. So this is a chromosome that's been deleted in a gene that causes syndromes, either Angelman syndrome or Prader-Willi syndrome, and these have very different, distinct pattern of abnormalities for children that have this finding. And it's unique in that the deletion can occur on the chromosome inherited from the mother or from the father. And if the deletion's on the mom's chromosome, we end up with one phenotype of Prader-Willi syndrome. If it's on the father's, then we end up with Angelman syndrome. The syndromes look nothing alike, but it's the exact same region that is lost. And it's because certain genes on chromosome 15 are only active on the chromosome inherited from your father, and other genes in that region are only active in the chromosome inherited from your mother. So depending on which chromosome we're looking at, you get a completely different pattern of problems for the patient. So FISH analysis has certain advantages. Again, when we do FISH, we're not looking at the whole genome like we're doing with chromosomes or other techniques. We're looking at one region, and often it's just one gene that you're looking at. So it's very specific. We're looking at about 100 kilobases, so 100,000 bases, as opposed to when we're looking at G-bands, we're looking at millions of bases for us to be able to pick up a gain or a loss. Its biggest application is in microdeletions and microduplications. So microdeletions are these small losses, as we saw with Angelman syndrome, Prader-Willi syndrome. Sometimes we can, on really good chromosomes, we can see that that region looks a little bit diminished, but it's really the FISH analysis that makes the diagnosis. And as we have regions that are prone to microdeletion, they're also prone to microduplication. So there's known sort of what's referred to as genomic architecture in the DNA sequences of these particular regions. And there's some copy number repeats or sequences that are present in multiple copies. And because of that, when they align based on sequence homology, sometimes the chromosomes misalign, and we can get little areas clipped out or little areas duplicated. So microdeletion, microduplication syndromes are known to be in particular regions of a number of chromosomes based on their sequence in the DNA. The disadvantages of FISH are, number one, the same thing that is its advantage, is it's highly specific. So if you use a probe, say I said, okay, I want to probe for possible Prader-Willi syndrome on this patient because the abnormalities that the doctor told me about look kind of like it could be that syndrome. And I run that probe and it looks normal. And I didn't learn anything because it's only going to look at one gene at one time. So this patient might have had something completely different, and if we don't look everywhere, we'll not know, we'll not find out what the problem is. It'll only look at the particular gene that you're testing for. So in order to look at the kind of resolution you get with a karyotype, you would have to run really 1,000 probes or more to look at all the regions that we do when we do a chromosome analysis. Oops. Okay. So being able to detect a microdeletion is always a challenge for us in the laboratory. Sometimes chromosomes don't grow as well as they should. The quality is not as good as you would like all the time. Sometimes they don't open up so that the metaphase is easily analyzable. And there's a lot of detail to what we do, and you really have to constantly be focused on every region of the chromosome, because for us, when we're doing a chromosome analysis, we often don't know what we're looking for. We're just looking at every chromosome pair, going through band by band to making sure that the two homologs look the same. This is a particular finding. It's hard to see it projected here, but it's a deletion on the very tip of chromosome 1. So you can see the arrow, the little black arrow up at the top here, is pointing to the very tip of this very pale region of chromosome 1. And it turns out that deletion of what's referred to deletion 1p36, because that's the band that it occurs at, turns out to be a very common finding, and we were missing it for years because it's very tiny and it's in a region that is, as you look up here, white on white. So the tip of the chromosome is extremely pale, and the background that you're looking at is the same color. So the background for our metaphase spreads is pale white, and the tip of this chromosome is pale white. So it doesn't grab your attention very easily. But new molecular studies with microarray analysis have shown that this actually is a more common deletion than we ever knew. Okay, here's just a second cell showing the same thing, and you can just see that, it is difficult to pick it up. Okay, so we have again a FISH probe that is now available to us, that if we suspect this abnormality, we can run the probe and verify the region is present or not present. And if you look at the two chromosomes in the center here, right here is the tip of chromosome 1 lighting up with the yellow probe. That shows that it's there. And then sort of an aqua probe further down the chromosome that is our control. So we know we're looking at chromosome 1, and you can really tell by size, actually. And we can see that the end of the chromosome picks up the probe. It's hybridizing, it's intact, it's not missing. If you look at the large chromosome right next to it, right here is the other chromosome 1, and you can see the sort of aqua control probe is lighting up, but the tip of the chromosome has no fluorescence on it whatsoever, and it's deleted. It's still a bit of a challenge looking at this prenatally. There are no structural abnormalities identified in fetuses with this deletion, and so there's no warning from the clinician that, oh, you should look for this because we found such and such when we had ultrasound performed on this baby. So we really have to be suspicious of it in the G bands prenatally for us to go ahead and put the probe on and look for it. In cases where we're diagnosing this in an infant or a young adolescent or child, there is known abnormalities that do pretty much describe the syndrome, so we have a better handle to know that we should be looking for that. Okay, so other advances in molecular cytogenetics have come about to improve our ability to detect smaller changes. We know that G-banding alone is insufficient to identify all the clinically significant microdeletions and duplications that are out there, and we've known this for some time. Sometimes when we get really good resolution and really clean banding, we can pick up really small things, but there certainly is no guarantee that you're going to always have that high quality of a preparation, or that you'll be able to see everything that you need to. We have now additional molecular cytogenetic technologies. They offer a more whole genome approach to find these very small submicroscopic imbalances. So the technology that is now being made more and more available to clinicians and for us in the laboratory is referred to as microarray analysis, or array-based comparative genomic hybridization. And this has been, past 5 years, pretty common to resort to this. If the karyotypes are normal and the fish doesn't show us anything, then we're going to go to more sophisticated technology and look at the whole genome at a molecular level. And of course, this doesn't involve a microscope. It's a completely different approach, but it is referred to as molecular cytogenetics. And it basically detects copy number changes, so gains and losses. It works via a genome-wide scan. It is in some ways less labor-intensive. The platforms or the equipment and instruments that perform this analysis are extremely expensive. The reagents are extremely expensive, and they're one-time use. In addition to the cost of the chips and the analyzer, there's a lot of controls that have to be run with these, because it's not a visual analysis, so you have to make sure that everything is working properly in your study for your patient. And so we run a control every time we run a patient. If you have more than one patient, you can batch them, but there has to be proper controls done, because you're not visually analyzing anything. It's extracted DNA, it goes into the instrument reads it out, and then you look at the result, but you have to run controls with it as well. Okay, so the objective is, it's an objective method to compare, as opposed to what we do when we do visual analysis. So this is done by an instrumentation. Okay, so what happens in array CGH, as it's called, is that we take DNA from a control population, so it's a normal control. We take DNA from our patient, we tag them with different colored fluorochromes again. So one will tag with red and one will tag with green. And then we take the DNA and allow it to bind to known probes. So instead of taking labeled fish and attaching it onto chromosomes, we're digesting the DNA from the chromosomes, tagging those with fluorochromes, and letting them hybridize to the fish probes on a slide. And in that fashion, they will compete to bind to these probe sequences. And if you have a deletion in your patient, it's not going to compete as well to bind onto that DNA, and it will show up as lower signal or lower fluorescence with whatever color we tag the patient with. And vice versa, if we have a duplication or an extra copy of a gene in our patient, it's going to compete better to stick onto those probes, and its color or its fluorochrome will be present at a higher amount. So red and green in equal amount give you a yellow signal. If we have extra of the green patient DNA, it's going to give you the green. If we have less, then the control red signal will show. So deletions show with red, duplications in green, and equal balance yellow. And so they hybridize to depending on how many probes you stick on. The high density ones are 60 to 100,000 probes in multiple copy. And the instrument will read each one of these spots and determine the amount of fluorescence for those particular colors that are present. Okay, so this just shows the two different samples, the patient DNA. Is there like a little focus on here? Does that look blurry to you, or is it just me?
No, I think we're both getting old and losing our...
Yeah, it might be. Thanks, Paul.
No, I need my reading glasses. Let's see. I can't tell.
Usually, it's just something you
it's
turn here, but
usually
it's not there.
yeah.
Okay, well, that's all right.
What does this say? I can't I don't think there's anything. There must be a way, but...
The alternate is to move the card,
see.
but
Is that better or worse? I'm not
I
sure.
can't tell. Okay, well,
It's
anyway.
the machine, it's not our eyes, I promise. So, sorry about that. We'll try to look at that.
So here just shows the two different vials of extracted DNA from the patient and the control, labeled with the two different colors that we allow to hybridize, and then there's a reader in the instrument that goes from each spot and determines what color of hybridization we see.
Yeah, hang on 1 second, please.
Okay. So there are a number of different kinds of arrays that are run. One is referred to as a back array, and these are only differentiated by the size of the DNA probe that's bound onto the chip. And back stands for bacterial artificial chromosome. So they're big probes. More and more we're going to very small probes. So back probes may be 1 to 2,000 base pairs of DNA sequence, and now we're doing them more 25 to 50 base pairs. So we're looking at very small probes nowadays, very high density, overlapping, and able to pick up small changes. And it's not unheard of to be able to pick up a 50,000 base pair, a 50 kb loss or duplication with a microarray. Okay, so methodology, we isolate the genomic DNA from a sample. The DNA is digested or broken up by enzymes. We label the patient and control samples with different colors. They are allowed to hybridize or to bind onto the probes on the microarray chip. We wash off whatever didn't bind, and then they're put into the instrument for the assay scanning and data analysis. Okay. So the resolution depends on the size of the probes and how far apart they're spaced. Really now they're using high density, high resolution arrays for the most part. The limitation to this particular technology is it can only detect unbalanced abnormalities. So balanced translocation carriers come up with a perfectly normal array. We cannot pick up reciprocal translocations with this technology. And lastly, it creates what's referred to as a molecular karyotype. And so what the instrumentation does with the data is it plots it alongside a chromosome, and it shows any deflection from what would be equal number or equal binding between the two different labeled control and patient DNA. And it plots deflections that would be the equivalent of a deletion or a duplication. And this is a copy of one particular patient sample with a deletion. Cytoscan is the name of the molecular cytogenetic array from a particular company called Affymetrix. That's the one we have in our laboratory. If you look at the sort of purpley blue spots along the top, these are all, each spot represents the hybridization from one probe. And so you can see that there's really, really a lot of probes that are being looked at. And in this level, right along this line would be equal copy number, so equal binding between the two probes. And then we get a sudden drop from right along this interface here all the way to this interface, where the intensity is lower than you would expect for equal binding, and it is actually a deletion, and this is Duchenne's muscular dystrophy deletion that's picked up with this particular study on chromosome X. And you can see that, even to an untrained eye, it's easy to look across and say, whoa, something's wrong. The line is deflected and it's not normal. Okay, so array formats can be either lower resolution, and why would we want to run low resolution when high resolution's out there? And there's the caveat that don't ask a question if you don't want to know the answer. Well, this is kind of don't ask the question if you don't know what the answer means. So when we're doing microarray analysis, for instance, in a prenatal setting, we don't want to find something that we have no idea if it means anything. So the idea is to set your resolution at a level where you can interpret the results in a meaningful manner, because we don't want to provide no information to patients. We don't want to provide misleading information or uninterpretable information in a prenatal setting where you really have no opportunity to examine the baby. So most of the prenatal arrays are looking at cutoffs around 250,000, 250 kb. Anything below that or smaller than that, we're really not going to look at. And they highly focus on regions where there are known syndromes that have been identified in other patients. So we want to be able to provide information that is interpretable in those settings, because you really don't have the opportunity of examining the patient. Okay, so we want to focus on regions where there are known microdeletions, duplications, and the end of the chromosomes, because the ends of the chromosomes are most frequently involved in losses and gains and changes. Okay. Kinds of indications for running arrays in a postnatal setting, infants born with multiple anomalies, cases of developmental delay or mental retardation of unknown origin, meaning they've already had karyotype analysis, perhaps there's been FISH studies, and we still don't have an explanation for this particular patient. Autism has been looked at quite a bit, and there have been a number of microarray findings that have been specific for patients with autism. And again, any individual with a suspected chromosomal imbalance, even if there's a normal karyotype, because as I said, we can't see everything on chromosomes. Okay, that's just an example of a particular one. Okay. When you're setting up these assays in the lab, each lab has to do their own validations, and you have to really set what your lower limit is that you're going to pick up as a loss or a gain. The way the testing is being performed today still is that if you pick up an imbalance or a copy number variant on a microarray, we will require confirmation with some other methodology. Now, most labs are using a FISH study to confirm it. So say we pick up a loss on chromosome 20, and that's what the array is telling us. What we're going to do is go back, grow cells, get metaphase chromosomes, find the FISH probe that identifies the region that the microarray tells us is missing, run it on the chromosomes, and look at the FISH study and make sure that that FISH analysis agrees with the microarray result. So we're doing confirmatory studies either by FISH, or some are doing it by PCR as well. So either way, but the recommendation still exists to confirm all these studies. Okay, and I'm gonna, okay. So high resolution formats, again, we're looking at probably 50 to 75 kb. Sometimes you can go a little lower if it's a region where there's a known abnormality and you have phenotype to go with it. Oligonucleotides are quite small, usually around 50 base pairs. Most of the arrays use about 50,000 to 100,000 probes in a study, and in each patient's analysis, sort of the downside of this is we discover what are referred to as polymorphisms. So changes that are unique to that patient, or maybe they've been reported in other normal patients, but they're not associated with abnormality. So it's, for every study you do, you're going to find things that you have to research to see if they've ever been reported as an abnormality, but also to see if they've been reported as an innocuous finding in other members of the population. Okay, and I'm about the end of my time, so I'm going to stop right here. And thank you for your attention, and see if you have questions.
I did.
Yes?
I had a question about when you're looking, a couple of patients that you had under the telomeres.
Uh-huh.
What significance do they play?
So the telomeres are the ends of the chromosome. So the top and bottom of each chromosome has a repetitive sequence referred to as a telomere that protects the ends of the chromosomes from falling apart, from deteriorating and degrading. As we get older, those telomeres shorten. You probably read articles about telomere shortening and longevity or lack of longevity. Just within the telomere region is a region referred to as the subtelomere, and that is the first unique coding sequence on each chromosome that identifies it as part of that chromosome. The telomere repetitive sequence are the same for the ends of all the chromosomes. Subtelomere regions are regions of focus for imbalance, so for deletion or for duplication, just because they're the ends of the chromosomes and they're usually what's going to be messed up if there's a structural rearrangement happening there. Okay? Anything else? Yes?
My son's analysis was done in 1991. Is there any benefit to doing it again?
To doing a microarray, definitely. Yes, because frankly, there'd be a benefit for anyone having an imbalance discovered on chromosomes to move on to microarray, because we can really define what region is lost or duplicated. So yes, the microarray study definitely, especially now with the high resolution studies that are out there.
Do we ask for that with our geneticists?
Yeah,
Actually,
sure, you can speak with them about it.
That's a great transition, because I forgot to mention Dr. Mattina, who's here from Italy, is going to be doing blood draws if you're interested, and is ready to do assessments starting right now, actually. Dr. Mattina has probably seen more children with 11q deletions than anybody in the world, and this is about, correct me if I'm wrong, about a $2,000 test, and you can get it done today or in the next few days through Dr. Mattina for no charge. Insurance sometimes covers it, but oftentimes does not, so I would strongly encourage you to take advantage of that, and there's a sign-up form right outside the room here, so please do that. We learned so much, and just kind of along those lines to answer your question, and you'll hear a lot of talks in the next few days about this, where we can determine exactly which genes now are missing, and we can then make correlations between which genes are missing and the kinds of problems your children have to identify the genes that cause these various problems. So,
Is that something that could potentially tell us why there's such a broad spectrum between people
Absolutely.
like Q2, such
Yes.
a wide variety of cardiac defects?
yes. And then another reason why Dr. Mattina wants to draw blood is there's an even newer technology out there to do DNA sequencing that might help us to understand why two children with the same 11q deletion may still be different, let's say, with respect to whether or not they have a heart defect. That's called deep sequencing. And so even if your child has already had the microarray done, I would encourage you to participate with Dr. Mattina to have the blood draw done so we can do this deep sequencing or new generation sequencing that might help us to begin to answer those questions. That was a perfect setup.
Is there any reason for, if you have the karyotype done on the parents, is there a reason for them to have additional? Gaining the insight?
Well, I'll just put it that a microarray will not pick up something in a balanced carrier. However, if your child has had a microarray analysis, it'll give more insight as to what, if anything, the parent would be at risk for.
Not parent at risk for, but if we could find a link, if there's something.
So knowing what the abnormality is in an affected child helps determine whether you need additional studies in the parent. Okay, anything else? Okay, well, thank you very much.
Here's another little